Okay, so it's 10 a.m. Pacific time now. Let's get started. Welcome everyone to the Astatine-211 DOE Isotope User Group Meeting. My name is Yawen Li. I'm the point of contact for the University of Washington Isotope Program. Today we have a great panel of speakers. So first, Rob Emery from the University of Washington. Rob, could you please wave your hand to the audience? Yeah, thank you. And then we have Lauren McIntosh from the Texas A&M University. Lauren? Hi. And Bob Mach from the University of Pennsylvania.
Hello, everyone.
And Brenda Sandmaier from the Fred Hutch Cancer Center and the University of Washington.
Hello.
We have Dave Eve from Ionetix Corporation.
Hello.
Okay. And each panelist will give a 15-minute presentation. Then the panelists will take questions from the audience at the end during the 30-minute Q&A session. Please feel free to ask questions using the Q&A button. Please note we recommend using the Q&A button instead of the chat for questions. And I want to remind everybody this meeting is being recorded. So from there, I will share my screen and begin my presentation. So in the US, the interest in Astatine-211 is increasing, but there are some significant hurdles, including high cost, that must be overcome. Currently, there is no countrywide effort for Astatine-211 basic chemistry or radiopharmaceutical development. The US Department of Energy Isotope Program is providing funding to increase availability of Astatine-211, but no biological studies are allowed under that funding. The NIH provides funding focused on disease treatment that uses current methods of labeling, making it difficult for researchers to look at basic chemistry. There is increasing interest by companies, but some want to see clinical responses before they will invest. To our knowledge, these companies generally have no concern about adequate production. There are 5 ongoing early phase Astatine-211 clinical trials in the US. They are all being conducted in Seattle at the University of Washington and the Fred Hutch Cancer Center. There are 3 phase 1, phase 2 clinical trials using Astatine-211 labeled anti-CD45 antibody conjugate BC8B10. This reagent is being evaluated as part of hematopoietic cell transplant conditioning regimens for patients with high-risk leukemia or myeloid dysplastic syndrome or non-malignant diseases. A total of 59 patients have been treated as of August 2022. Dr. Sandmaier will give a talk on the early results later today. I'm looking forward to her presentation. Recently, 2 new phase 1 clinical trials have been approved and are coming up for the treatment of multiple myeloma. In the US, to the best of our knowledge, Duke University and the University of Pennsylvania have been in the process of transitioning their Astatine-211 reagents into clinical trials. Outside of the US, the Osaka University in Japan has started a phase 1 clinical trial to evaluate sodium acetatide for the treatment of thyroid cancer. This trial plans to enroll 11 patients. We believe the Fukushima Medical University in Fukushima, Japan, has started a phase 1 trial to evaluate the safety of using Astatine-211 labeled meta-acetylbenzoguanidine MABG for the treatment of neuroblastomas. The information on this trial could not be found on clinicaltrials.gov, but according to a colleague in Japan, we believe the MABG clinical study has started. About 10 years ago, the research groups in Gothenburg, Sweden completed a phase 1 clinical trial that evaluated Astatine-211 targeted alpha therapy for ovarian cancer. This dose escalation study suggested direct administration of Astatine-211 labeled MX35 antibody Fab2 fragment into the peritoneal cavity could be a safe method for the treatment of metastatic ovarian cancer. The Gothenburg group is planning for a phase 2 study in the near future. At the University of Washington, Astatine-211 is produced on the 50 MeV multi-particle variable energy cyclotron at our medical cyclotron facility via the alpha-2N reaction from bismuth metal targets. We now have 2 different setups for producing astatine. The first setup has been used for over 20 years. There are a couple of photos here to show the bismuth target and target station. This target was developed in collaboration with Triumph. The target is irradiated at a 10-degree angle. To make this target, bismuth metal is melted onto aluminum target bodies, then machined to the desired thickness. This target has a large bismuth surface, about 12 centimeters by 2 centimeters. It has a fully stopping thickness and contains about 4.25 grams of bismuth. To obtain a higher production rate, we use a 29 MeV alpha beam, and no Astatine-210 is observed based on HPGE analysis. With an average electrical beam current of 50 microamp, about 26 millicurie is produced in 45 minutes. We routinely produce 130 to 150 millicuries in 4 to 5 hours for clinical studies using this setup. And thanks to the effort led by the UW Medical Cyclotron team, we now have a new astatine target and a new target station, the design of which is available for DOE University Isotope Network to use. The new target is designed to withstand 100 microamp of 29 MeV alpha, be compatible with commercial remote retrieval system, contain target housing materials that do not interfere with chemical processing, and minimize target housing activity for safety handling, safe handling, and minimize long-lived radioactive waste. The target station is designed to have ability to remotely load, irradiate, and retrieve targets. To transfer irradiated targets into shielded pegs or pneumatic target transfer systems connected to hot cells, and to accommodate target material in various forms. Rob Emery will discuss the new target and target station in detail in the following presentation. To separate Astatine-211 from the irradiated targets, a wet chemistry isolation process is conducted. It begins by dissolving the target in concentrated nitric acid. Then the nitric acid is distilled away, leaving bismuth salts containing astatine. Then the bismuth salts and astatine are redissolved in 8 molar HCl. After everything is completely dissolved, diisopropyl ether, DIPE, is used to extract astatine. The DIPE is then washed 4 times with 8 molar HCl to remove residual bismuth. Finally, the astatine is back extracted into sodium hydroxide and transferred into a conical vial. The sodium hydroxide is neutralized to pH 6.5 to 7 before antibody labeling. The runtime of this manual process is typically 2.5 hours, and the non-decay corrected yield is about 60%. If you would like some astatine for your research project, please visit the NIDC website, isotopes.gov, for quotes and for placing orders. We have 15 millicurie and 50 millicurie batch sizes. After overnight shipment, you can expect about 10% of shipped quantity at receipt due to the short half-life. We ship astatine in near neutral pH in a plastic V-bottom vial. the total volume is typically 0.6 to 0.8 milliliter and is always less than 1 milliliter. For overnight shipping, we send through FedEx. But if your institution is within driving distance from Seattle, a local courier can be arranged. We have developed a semi-automated Astatine isolation process using tellurium-packed columns. Briefly, using this process, 10 molar nitric acid is pumped through an inline dissolution chamber to dissolve the irradiated target. Then a solution of hydroxyamine hydrochloride is added to the astatine bismuth mixture to reduce nitrate concentration. Then the solution is adjusted to about 1.5 molar HCl by adding 8 molar HCl. The prepared solution is loaded onto a tellurium column in 1.5 molar HCl at a high flow rate of 6 milliliter per minute. Astatine is rapidly absorbed by the column. The column is rinsed with HCl and water subsequently, and finally the astatine product is eluted in 1 normal sodium hydroxide. This method eliminated the nitric acid distillation step and shortened the overall runtime from 2.5 hours to about 1.5 hours. However, the final product contains a little bit of tellurium impurity, about 20 to 50 ppm. The product might have breakthrough hydroxyamine. The non-decay corrected yield is about 90%, and the radiochemical purity is typically greater than 99%. The final product is in about 1 milliliter of 1 normal sodium hydroxide. The astatine obtained using the tellurium column method is suitable for labeling of closodecaborate conjugated antibodies without added oxidant, providing labeling yields of 70 to 80%. We have optimized the quantities of reagents to prevent hydroxyamine breakthrough, and we have developed a quality control method for detecting hydroxyamine. This HCl method utilizes the rapid reaction between hydroxyamine and anhydride at room temperature and provides a detection limit lower than 0.05 microgram, which is well below the level known to have any toxicity effect. We have been producing on average 2.2 curies of astatine per year since 2018. In 2021, 4% of the produced astatine was shipped to other US investigators through the NIDC. 20% was used for chemistry and preclinical studies at the UW and the Fred Hutch. 76% was used for the 3 ongoing clinical trials. It has been a privilege to be able to work with all the Fred Hutch and the UW researchers on developing new astatine radioimmunotherapy. These are the PIs of the research groups that we have collaborations with. Here, I want to thank them for their tremendous effort in advancing astatine radioimmunotherapy. These are the collaborative research areas that we are working on, including non-myeloablative stem cell transplantation, cell and gene therapy for non-malignant blood disorders, latent HIV infected cells, radioimmunotherapy for lymphoma, leukemias, and multiple myeloma, radioimmunotherapy to study graft versus host disease, radioimmunotherapy with other novel agents for multiple myeloma, radioimmunotherapy to treat hepatocellular carcinoma. With that, I would like to thank everyone in the radiochemistry group and thank you for your attention. We will hold questions to the end. Now let's move on to the next speaker, Rob Emery, Director of the UW Medical Cyclotron Facility. Rob, please.
Yeah, thank you, Yawen. Thank you for the talk. Let me share my screen here. Okay. So yeah, today my name is Robert Emery, and I'll be talking more about the the radiation side of the target and the target tree itself and what we've developed in the last few years and where we're trying to go with that. So the accelerator that we use here is a Scanatron's MC50, commissioned in the early 80s. It was developed to do protons and deuterons, but it's also capable of doing alphas. It just wasn't optimized for it. And so over the last, 30 years, we've really been working on the accelerator and the beam-like components to generate an alpha beam we could use for Astatine clinical trials. It's a variable energy machine. Back in 90, which is a little before my time, we got out 1 microamp at 29 MeV. And today we're getting up to 50 microamps with a lot of work and tweaking. And just to show you a little bit about that, I won't necessarily go into all the modifications we've done, but the majority of it had to do with the ion source and then the extraction of the beam. We have a cold cathode source, and that is coupled to our RF system, which also acts as a beam extractor from the source. And so that tightly coupled system, with that, it was a little bit difficult to isolate where our issues were. So we did a lot of work with our source, our D-tips, which is the extractor for the source, and the source window and that type of thing. Once we started getting a significant amount of beam out of the machine, we couldn't get it out of the extractor. So then a significant amount of work was put into modifying the extractor, reducing some of the scrapers, adding some more water cooling. And then finally, once we got the beam out of the machine, we couldn't get it to the target. So we helped that out with some larger quadrupoles. So it was really a lot of work over a lot of years to get this to go. Our previous and somewhat current target station was this the station that we collaborated with Triumph to install. Some of the downsides of it are it is a larger target, and it makes it difficult to operate with a remote retrieval system, and so there's some additional dose taken on for manual retrieval, and then just sort of the limited flexibility of the station. We really solely use this for our astatine and then tin production. It really doesn't lend itself easily to other sort of experimental type coin targets. And so a few years ago, we went about designing a new target system with the criteria that Yawen briefly talked about. The ability to run 100 microamps at 29 MeV alpha. That was sort of arbitrary. We're 50 now, maybe somebody will get to 100, but that was a design criteria. The ability to auto load and eject targets to minimize exposure. Compatible with the commercial transport system, adaptable for other isotope or research targets, a cost-effective system. But really sort of the biggest part of it was the ability to share this system with other DOE IP production sites. And I'll just take a second to say here that the Department of Energy Isotope Program is really, I think they've really put a lot of time and effort into encouraging and driving their facilities to share information and bring products like Astatine-211 to the country to be used for these clinical trials. So I applaud their effort there. So we went about this starting with some thermal modeling, designed the water cooling system. We're using ANSYS, space claim and fluent, and then MCMP for the energy deposition model. Things we came across in the design were the most important things were really the lateral energy distribution and the water flow rate. And so by playing with those elements in the cooling design, we were able to come up with a design small to fit into a commercial target system. Our goal was for a 1 inch, or I'm sorry, a 1 inch in diameter, 6 millimeter thick target, no bigger than that. Here's an example of where it didn't work. These are some of our earlier target designs where we're not using thermal modeling. We were just using sort of best guess. And you can see with this poor water flow model, we ended up with some hot spots and then actual target failure. And that was an older target design that we had not modeled for, that we went back and looked at to try to figure out why it failed. With our new design, we get a much more even distribution of the water flow, and you can see the top picture is a combined water flow heat map. And then you can see the bottom two pictures with the water flow on the left and the heat map is much more even distribution. And then theoretically allows us to get to that 100 microamps. This is the new target design. We're using aluminum backing to make it compatible with the chemistry on the backside. The bismuth thickness we optimized to capture just the range of energy that's going to produce the astatine. Now, bismuth is a very poor thermal conductor, and so as much of the beam as we could dump into the aluminum, the better. On the bad side of that is we create a radioactivity in the aluminum we don't want. So we're currently using a 5N aluminum backing to reduce that. In the future, we'd like to look into go into a 6061 aluminum backing. It's a little cheaper and easier to work with, and then have the top of that plated with gold, and then bismuth on top of that. We're also looking into electroplating the bismuth, and these aren't necessarily required steps, but it's just our investigation to create a system that will best suit different sites around the country. We do plan on having a remote retrieval system in the near future, and so coming up, this might not be as important for us, but other sites that don't have that remote retrieval, it might be useful. We do have some special tooling to deliver a precise and even bismuth layer, and that was important because we're really trying to nail down the appropriate thickness and tolerance for the bismuth layer, and so to get it tight in the beginning was important. And then we use a profilometer to confirm the thickness and the flatness. You can see down the lower left, those are some first cuts at doing some gold electroplating. And then this is the end result. It's a target station that was mechanically mostly designed by our engineer Bob Smith, and it's really kind of a nice system. You can see here's most of the pieces and parts. The gold valves aren't there, but we really designed it for sort of a do-it-yourself kit to put together. Most of this equipment is off the shelf from McMaster or wherever, and there's a little bit of machining, but it's really pretty straightforward. Where we are with the system now, we've done 11 production runs to date. We're continuing to ramp up the beam power. I think we've run up to 35 microamps on this target station. So we're getting pretty close to our target goal of 50. We have not seen any issues or anything close to failure. We're evaluating the make rates, ensuring we have the right things of bismuth, the personnel exposure, the chemistry yield. That was the one concern from our chemistry group, to make sure that we're getting a similar yield. With a shorter half-life, we can only produce so much. And then we're using some novel beam scanners from a company called DPace to dial in our beam profile, which is very important for the heat load distribution. So those are the things we're currently looking into. We're currently only producing astatine on the new target station for target evaluation or radiochemistry, but we would soon like to move to the animal and clinical trial production once we optimize the system, not only on the targetry side, but also on the radiochemistry side. They're having to adjust their their chemical process a little bit for the smaller volumes of solution. You can see here we've designed, or Bob Smith designed along with Don Hamlin and produced a smaller dissolution chamber. So we're really trying to come at it as a holistic project. And then where we're going in the future, we're bringing a new beamline on, and we've received the equipment and should have it fully operational by spring 2023. We will produce a new target station for this, one that we modify a little bit to make some improvements on lessons learned. We're going to add a thermocouple for real-time temperature monitoring, and also to validate our thermal models. We'll add a new force sector collimator that one of our engineers, Eric Dorman, designed, just to give us a little bit of additional information on where our beam is going. We'd like to add some air sampling and radiation monitoring right at the station, and then a modified target backing to reduce exposure. And then finally, we plan on installing a target transfer system, I imagine sometime within the next year. Thank you.
Thank you, Rob. I would just remind everyone, please enter your questions using the Q&A button. Now, I will invite our next speaker, Lauren McIntosh. Please, Lauren.
Thank you. Let me pull my slides up. And share. All right, so hopefully you're seeing the slides. So I'm Lauren McIntosh. I work here at Texas A&M at the Cyclotron Institute, and I'm going to tell you about where we're at with our Astatine-211 research. So since 2021, sort of the highlights to kick us off, we've had quite a bit of beam development here. We were getting something like 8 microamps available overnight as of last year, and we're up to 12 to 14 pretty reliably overnight, and even more stable with our machine, which I'll talk about in a minute, which is different than the one that they're using at University of Washington. We are continuing to work on our targets as well. We've got a shallow well now, and I'll show you what's going on with that. And we are working on an automated system, which will be based upon the designs that you heard about from Rob. So we've got different time space constraints and things like that. So we're working on that. And we have also automated our dissolution and are able to send a dried column, which I'll talk about from our chemistry, to collaborators, as well as looking at some chemistry with astatine. So we're really more in the development, fundamental research area. We're not doing clinical trials here in Texas yet. So this is where I am. This is the Cyclotron Institute. We have a couple of different cyclotrons here, which are mostly used for basic nuclear physics research. So we have a K500, and we have a K150, which is our workhorse for astatine production. So here in the K150 vault, it's an 88-inch cyclotron, which might be familiar to some of you. And we've got a beam line right here that sneaks in behind the horseshoe magnet in the cave where our cyclotron is housed. And so here's a nice shot of our K150, and we're using the same reaction, of course. We've got alpha beams at about 28.8 MeV on our bismuth target. And here is a photo of our target chamber here. And so our target is way in the back here, and then we've got some cooling off the back. So let me show you what that looks like. So we're using a coin target. We went to this early last year. So as many of you know, if you're interested in astatine, that the melting point of bismuth is pretty low and the boiling point of astatine is fairly close. So we are able to use the low melting point of bismuth when we're making our targets, and then we want to keep them very cool so they don't melt again. So currently our process involves a hot plate with some pellets and then an ultrasonic soldering iron, which goes across the entire surface of our coin targets here. So we are using about 1 gram of bismuth in these targets, going for the same thing that was talked about from University of Washington, where we want to make sure that all of the bismuth that we have in front of our alpha particles are capable of making astatine. So the alpha particles are losing energy as they go through the bismuth, and at a certain point it's not going to create astatine anymore, and at that point you're just adding heat. So we're allowing that to go into our aluminum backing. And we have a directly water cooled target as well. So our target goes here, and this is the flange in the back, so it was on the back edge there of our target chamber. Water comes in here in the center and then goes out around the sides, cooling our target down fairly well. So that's what we're working with right now. We've also made some improvements in our current, and I just want to show this off a little bit quickly. So this is towards the beginning of when we were working on creating Astatine-211, and we had a current that was around like 5 microamps given what we're measuring. So this is not electron suppressed. This is not a official Faraday cup. This is pulling electrons off the back of the target. And we had things kind of sort of declining over time. As of last summer, and we would measure something like 14 to 13 using that measurement, right, which is not an absolute measurement of current, I want to say again. And as of this summer, we were closer to 20 in that realm. So we irradiate overnight. We don't have as many alpha particles available in our beam as you do at University of Washington, but we are able to produce some sufficient amounts of Astatine-211. So this is looking at the progress that we've been making over the last few years. So we started this iteration of Astatine-211 production in 2019, and we were able to get a beam current of something like 4 microamps, producing about 24 millicuries of Astatine-211, which is definitely enough to do some fundamental chemistry with. but we've made several improvements, working on the source, working on the injector, figuring out where the deflector needs to be, all of these important things for accelerating an alpha beam. And over the past few years, we've been increasing the things that are really the most exciting, or our highest average beam current, because that tells us really what we're going to be able to make and what our target can withstand. So we've gone from something like 3 all the way up to 12 this summer, and we've gone from this original 24 millicuries up to making pretty regularly about 100, which has been really great. We're learning a lot about astatine here and able to develop some of our next methods in our Astatine-211. Our target extraction currently is being done by hand, but we are working on an automated system. As I mentioned, it's not, I don't have pictures because they're still putting stuff together. It's based on drawings we were able to get from the University of Washington, and talking to some of their engineers about what they're doing with their coin target and their automatic target setup and retrieval. So we've been working on that, and we do have to make sure that things are radiation hard enough, because our target system does go in the vault with our cyclotron. So we have a lot of neutron flux there, and we need to make sure that things are going to work automatically when they're in that sort of an environment. The other thing that we've been working on, so Evgeny Trashetov has done a lot of work on automating our target dissolution. So we also dissolve our target in nitric acid to do a liquid extraction of our astatine. And he's set up, we have this set of pumps and valves over here. So this is our target chamber. So the target, our little coin target, goes right here, and the lid comes down. There's an O-ring, it connects directly to the surface. And so we can get a good connection, have liquid directly on the surface of our bismuth target that's been irradiated and made a bunch of astatine. The ADA here, the automated dissolution apparatus, will then pump nitric acid in, and then pump that, push that dissolution solution, as we call it, after about 10 minutes. That's about how long it takes to dissolve a target. This dissolution solution will get pumped out and into a dissolution vial, so that then we'll have none of the products that get made on the aluminum, as Rob alluded to, but just the products, the astatine itself that's been produced on that bismuth target. And this dissolution vial, we can then look at how much we made there without having the background of things that might have been made on their aluminum frame, and that dissolution solution is ready for separation on a column, which is the way that we separate out our Astatine-211 from our bismuth matrix. So we have 3-octanone on a little chromatography column, and the 3-octanone interacts with the astatine and separates it out. The bismuth goes straight through, and then we're able to elute our column using ethanol and to pull the astatine right off. So we've got a graph about that here. In this particular one, the column sat for 3.5 hours after it was loaded. So we have our load solution here, and we've got our bismuth 207 coming straight off. And then we have a washing with water. Column sits for 3.5 hours, and we're able to elute with ethanol and get a very high yield, over 90% of our relative activity back off using this method. So from the target dissolution through pulling things off the column is something like 20 minutes, and time is very important when we're working with astatine because of the short half-life. And so that's been working rather well. Here's an example of everything we're able to get out. So on the left, we have our target spectrum on the HVGEs. These are just looking at the X-rays and gamma rays coming off. So we have quite a few things on our target at the beginning, and then after things have been separated out, we've got our astatine with its little bit of daughter polonium on its own. So that's been working well. And one of the other benefits of using this column system is that we've been able to send via a local courier a column of this to some collaborators that are working at the University of Texas MD Anderson Cancer Center. And so they've been able to use some of this astatine to do some early studies on how things might be able to be radio labeled. So that's really exciting that we've been able to get that collaboration moving forward even more in the past year. So another thing that we've been looking at, we have this column with the 3-octanone that works rather well for separating out astatine, but we're also interested in the fundamental chemistry of astatine here. And so a collaboration with Texas A&M and University of Alabama at Birmingham. So Jonathan Burns is a fairly new professor there. So he's been looking at how we could improve this and what sorts of interactions are actually going on in between the astatine and a ketone molecule. So this 3-octanone is the one that we've been using on our columns very successfully. And he was studying what we could do to increase the affinity for astatine and find out how the astatine is actually interacting. So we are pretty sure that the astatine is in an astatine monoxide form at this point when it's after it's been in this nitric acid. And so he was looking at the interactions with a few different molecules. So if we add a phenyl group here to our ketone, what does that do? So we see here, we've got our ketone molarity versus our D value for separating out the astatine. And we see that this added electron density that's available from this phenyl ring is actually increasing the affinity for our astatine and separating it better. So that's interesting. So what happens if we've got those electron donating groups, and what if we add another carbonyl to it? So if we go to this dibenzoyl methane, we were interested in whether we could get the 2 oxygens to actually interact with our astatine, and we see that these are mostly just on the same line. So that's very interesting. So why is that happening, and can we do anything about it? So the idea is that perhaps these carbonyls are actually not situated in a way that they can both interact with the astatine. So if we increase the length of that carbon bond and allow more of them to be able to interact, what does that do? So you see when we add a couple of carbon atoms in here, we're actually getting a much higher D value. And if we add another, we're getting even higher, up to, this is something like an order of magnitude increase, basically because energetically they can interact. So what can we learn about this from looking at some theory? So we have some DFT calculations with some collaborators here at Texas A&M, and we can see what we think is happening here. So we have a monodentate interaction. So the 3-octanone, the acetophenone, and the bibenzoyl methane are all doing this monodentate interaction. They have very similar delta G's here. And then as we add carbons to the chain and allow for more of these ketones to interact with our astatine monoxide, we actually see that it's energetically favored for both of the oxygens to interact with the astatine for these longer chains. And we'll be interested to see what else we can learn about the astatine interaction as far as fundamental chemistry goes, since astatine is a very not very much studied atom on the periodic table, since there are no stable forms. So that's interesting. We'll be continuing this work and other work in fundamental chemistry. The sort of short summary are that we're increasing our astatine production and working on automating lots of our systems so that we can handle this increased astatine production. We're sending astatine to collaborators at MD Anderson and talking to the Isotope Program about how we can make astatine available in our area through the NIDC in the future. And we continue to be interested in the fundamental chemistry of this interesting atom. So I want to thank our Astatine-211 team. So this is when we had some collaborators from UAB come down and help us out with a run this summer. So there are all these people that help make Astatine-211 and are interested in what it can do. I also want to make sure to thank our operations people. We have an excellent radiation safety team, as well as our accelerator physicist who gets all the beams out of the machine for us. I've got a couple of funding things here, thanks to the Isotope Program and all of the other people that are involved tangentially in supporting this effort. And I wanted to take a minute to advertise for coming to join our team here at Texas A&M. We're looking for a radiochemistry postdoc. So if you're interested in Astatine chemistry or have any experience with it, we'd be very interested to talk to you. You can reach out to me. And on a different note, we also have opportunities through the HIPPO program for undergraduate students. And we're the lead institution on this. And there are 17 different US institutions that are taking part in it, however, and we have opportunities and research at all of them. So I would encourage you to reach out to me if you have questions about that as well. So thank you.
Thank you, Lauren. That is a great presentation. I'm very excited to hear you guys are at the 100 millicurie level now. Congratulations. Okay, so again, I want to remind everyone to continue submit your questions using the Q&A button. And now let's move on to our next speaker, Bob Mach from the University of Pennsylvania. Bob, please.
Okay. Let's see, bear with me for a second. Thank you, Yawen. I thank you for the opportunity to present today. What I'm going to do is give an update of our activities over the past year in the area of Astatine-211 production in radiochemistry, different radiochemistry projects here at Penn. A couple things I need to point out to the group. Number one, that the person who used to give this presentation and previously was in charge of our Astatine-211 program was Dr. Mehran Meghbandi. Mehran basically led this program for about 6 years. And earlier this summer, he left Penn and joined a biotech company in North Carolina in Research Triangle Park area. So we're a program in transition right now. So I've stepped in for today to give this presentation, and I'm working with some of the people in the Cyclotron facility who worked with Mehran on our Astatine program, in particular, Dr. Yawen Li, our Cyclotron facility operations manager, and Paul Moderano, who is a chemist in our Cyclotron facility, and a lot of the work that I'm going to present today really was done by Paul. The other thing I'd like to point out is relative to the other presentations you just heard, ours is likely going to be a letdown because just to let you know that our Cyclotron facility for producing Astatine-211 is in the Department of Radiology, and our Department of Radiology has a very active operation and business in producing these standard radionuclides for imaging research. So we produce FDG for our clinical studies. We have a very, very active research program with fluorine-18 and carbon-11. So what you're going to see today is the reason for what you're going to hear today is a little bit different from the other presentations, is that the amount of space Astatine-211 production in radiochemistry has in our overall scheme of things is relatively minor relative to everything else is going on. But be that as it may, we believe Astatine-211 is an extremely important radionuclide. We do spend time trying to produce. Is why we have a dedicated cyclotron to produce Astatine-211, and we try to contribute in areas where we think are a little bit different from everyone else, what everyone else is doing, but and contributes to the field in general. And that's perhaps looking at, after you produce Astatine-211, how do you radio label molecules with it, and what does one do with it? So that's really what our focus has been on at Penn, is more applications based. So here's an update of some of the things that have been going on in the cyclotron facility since the past year. Let's just see, go to pointer options there. One of the things you're going to hear about is we have 2 radiopharmaceuticals that will be labeled with Astatine-211 that we'd like to take in the clinical trials. MABG, as you heard about a few minutes ago from Yawen's talk, which is currently being done in Japan. We'd like to do clinical trials with that at the University of Pennsylvania in collaboration with investigators at Children's Hospital of Philadelphia. And we have one of the tracers I developed. It's a PARP1 radio tracer that also looks very good in preclinical studies. And our plan is to try to take that into first in human clinical trials sometime within the next year or so. So in order to produce that in a facility, we installed these 2 isolators in our GMP facility. And the goal is to actually be able to produce Astatine-211 in a GMP environment for these clinical trials. We focus on small molecules. We think small molecules, the biological half-life of a small molecule is more compatible with the half-life of Astatine-211. So we think Astatine-211 is a really nice therapy radionuclide when it's incorporated in small molecules. And that's really what our focus is on at the University of Pennsylvania. In order to produce the radioisotopes here, we need a cyclotron to do so. You've heard Moran speak about this in the past. As a JSW-30 MeV cyclotron. It was installed in 1985, operational in 1986. And for many years, it was a proton and alpha accelerator. After we got our IBA cyclotron about 9 years ago, we recommissioned this into a dedicated proton-only accelerator for production of Astatine-211. We used to have a target changing station here that we removed, and we now have a single port. Here are the 3 target ports we have here that could accommodate the astatine targets. We are currently using the Y station for Astatine-211, and we have 2 open ports for testing out new targets. And I'll talk a little bit about that in a moment. Well, the reason why we want to test out new targets is that our current target is actually not great for running at higher currents. Our cyclotron can run between 5 and 20 microamps deposited on the target, but when we run at the higher beam current, we've been our target isn't designed to tolerate that, and we melt the target. So we are currently running on the low end of this, around 5 microamps, and we're able to produce between 5 to 6 millicuries in the target. And those in a moment, I'll talk about how we isolate it, and we get about 3 to 4 millicuries off the target using the system to the right here. But we're currently working with Sumitomo. They have provided us a new target that has more efficient cooling, and we're going to install it in one of the open ports, and hopefully we'll be able to increase the amount of beam on target so that we can produce higher levels of Astatine-211. I think we're a little bit low, below where we would like to be to support our clinical trials. So hopefully we can increase our production yields in the coming year using this new Sumitomo target. How we isolate Astatine-211 is different from the other. Let's see if I do that there. Yeah, the other programs. What we use is an induction heater here, and what an induction heater here has a high frequency AC current that's sent through an electromagnet, and what that does is through creating eddy currents in metals, it creates an ability to get focused heating of metals in a very short period of time. You can see when you first turn it on, the target aligns with the magnetic field as it flipped up here. And you can see it's getting very hot, very quickly. So we use the dry distillation method. You previously saw some small furnaces that we developed with a smaller footprint. We have actually switched to this induction heater, and we think it works very well. So that's how we're currently isolating Astatine-211 using this dry distillation method. We trap this in a sodium hydroxide solution, and it's suitable for radio labeling purposes. So one of the things we've been working on is how does one radio label small molecules with Astatine-211? And an area of radiochemistry that we've become very interested in recent years is this photoredox chemistry. And this is a paper that was published in Nature in January 2021. It's using a chemistry method that was developed by David McMillan at Princeton University. For those of you who may recognize that name, David McMillan received the Nobel Prize in chemistry in 2021, and for his very creative work in synthetic methods. And this is one of the methods that was developed by this lab, and it was used for introducing either carbon-11 or tritium in the form of a methyl group in the molecules that contain a bromine atom here. And here are the reaction conditions for doing that. Basically what you do is you have a photoreactor that shines blue light on your substrate, and with these various catalysts is able to, through a free radical reaction, to convert the bromine group into a methyl group. And it actually works quite well. Some of the people here at Penn, Alexander Schmitz and Sharon Lee, are on this paper for doing some carbon-11 work. And this is the photoreactor in our hot cell adjacent to our carbon-11 methyl iodide module. So we become quite intrigued with this type of radiochemistry, because it really does increase the ability the versatility in introducing carbon-11 into small molecules. Now, Jacob Ritter actually was able to show that you can use photoredox chemistry for a variety of different chemical reactions, and he showed that you could actually iodinate and chlorinate, halogenation reactions with this, using a leaving group here, which is a rather unusual leaving group. It's a thianthurium salt, which I'll show the structure of on the next slide. But this is a very versatile chemistry that can be used to do carbon-carbon bond coupling, or carbon other things bond coupling in a very efficient way using this photoredox chemistry. So we asked the question is, can we use photoredox chemistry to radio label things with Astatine-211? So to test that, what we did is here's a structure of this thianthurium salt. They're kind of tricky to make, but you can make them. And the first reaction conditions we did is put it on a number of different substrates, and labeled it with both iodine 125 and Astatine-211. Here are the reaction conditions here, and here's the catalyst we use here. And this is a setup that we use for introducing or doing radiochemistry with iodine and Astatine-211. You can see it's a very small apparatus. Again, this is just provides a very intense blue light for catalyzing these photoredox reactions. So here are some results for labeling with iodine 125. You see the yields are actually quite good across a variety of different substrates. They're high to fairly good, except for this one down here, where the yield was actually quite low. So on this slide, we were able to not only just replicate the work that Dr. Ritter did in halogenating molecules, in particular introducing iodine, but we're able to use it for introducing radioiodine, in this case, iodine 125. So that's great for iodine 125. What about Astatine-211? Well, here's the results for introducing Astatine-211. I'd like to point out that there are some subtle differences between iodine 125 and Astatine-211 that we noticed. By and large, the yields we got with iodine 125 were replicated with Astatine-211. There are a couple of exceptions. The red stars show the reactions where there was a much lower yield that we got with astatine than we did with iodine 125. Here, this is about 80% or 70%, I think. It's around 10% in this molecule here. And in this reaction, it didn't work at all, probably because of some rather significant steric hindrance properties here with this group out here, and astatine is a rather large molecule. However, we were surprised and pleasantly surprised to see that the radiolabeling yield of this molecule was much better with iodine. With iodine, it was around 10%. Here we got about 40%. So we like this method. We think it's a very versatile way of introducing astatine-211 in the molecules. There are a lot of other differences that we've noticed with the behavior of astatine versus iodine. I won't go into that now. This is still very much a work in progress, and I think in our next presentation we'll give you an update on what we're discovering with the use of this photoredox chemistry with astatine-211. But it is quite interesting, and it is I think a neat way of introducing radiolabels into small molecules. Other things going on at Penn. Well, as I mentioned, we have we'd like to start 2 clinical trials soon. The first one is MABG, and this is the neuroendocrine tumors. And we also want to do astatine-211, this PARP1, which we call parthanitine. The solid tumors, we're going to focus on breast, prostate, and ovarian, and also neuroendocrine tumors. And one of the things we plan on doing, and we've been talking about doing this for a while, but we're now trying to take steps to actually to accomplish this, is we've been discussing with Memorial Sloan Kettering in New York about actually shipping astatine-211 to their program. And how we will do this is we'll just ship a target to them. They're setting up the radiochemistry operation at Sloan Kettering for isolating the astatine-211 and doing radiolabeling studies. So we've been talking about being able to ship astatine outside of Penn for a few years now. We've been taking the steps in recent months to try to work that out. And as you can imagine, there's a bunch of headaches that you have to go through, and from regulatory and material transfer agreements between the 2 institutions to make this work. But we hope to be able to send some astatine-211 up to Sloan Kettering within the next 6 months. So that's really here are our future plans, to get these clinical trials up and running and to be able to ship astatine-211 off-site, in this case to Memorial Sloan Kettering. So that really ends my presentation. I'd just like to acknowledge a few people. Moran McVondy was the person who led our program. He really was a very creative scientist. He did some really neat stuff, and he's going to have a great career in the biotech company he's working from. But he did a lot of interesting things with astatine here, and we appreciate his everything he did for us. Paul has taken over a lot of the lab work for me. Worked very closely with Moran over the past few years. Sharon Lee, as I said, is our cyclotron facility manager, who works with our cyclotron engineers to try to keep the JSW up and running and produce astatine-211. And the funding, we have had funding from DOE. I think it recently expired. So now our current funding as about a month ago is NIH through this NIBIB grant for doing some new radiolabeling with astatine-211, and a study funded from the NCI to Dan Prima for looking at our radiolabeled PARP1 radio tracer in various animal models of cancer. So with that, I've completed my presentation, and I be happy to answer questions during the question and answer period.
Thank you very much, Bob. So now let's move on to our next presentation. Brenda, please.
Okay, can you see my slides?
Yes, I can.
And am I in presentation mode?
Yes, you are.
Okay. Okay, thank you. It's a great pleasure to be able to present to this group. Mine is a little bit different. It's very clinical, and it's served at the end user. And for me, it's very exciting to listen to all the preceding speakers to understand a little bit more about how we got where we are and where we're going at the production side. So I'm going to talk about the use of astatine in clinical trials for hematopoietic cell transplantation. And because I realize that not everybody here is a bone marrow transplant person or a hematologist, I'll give a couple of introductory slides about what transplantation is. Essentially, the candidates for transplantation are really patients where nothing else works. They're advanced leukemias, or myeloma, or otherwise incurable cancerous and sometimes non-cancerous marrow or blood disorders, including sickle cell disease. And the aim really is to replace diseased marrow in the patient with normal marrow from a normal, healthy donor. So typically, when we're preparing a patient for transplant, we give some sort of combination of chemotherapy and radiation. And really the function is to destroy diseased marrow and also to suppress the patient's immune cells so that the marrow graft will be accepted. Even when they're perfectly matched, the patient's immune system is competent to reject cells from a donor. And the healthy marrow graft essentially replaces the diseased marrow and also provides a function of what we call the graft versus leukemia effect, where the immune cells will also do a little bit of cleanup of disease and prevent relapse. And the donor has to be healthy, and they could be matched, related, unrelated, and they can be partially mismatched, such as we call haploidentical or cord bloods, in which case we need more immune suppression so the patient doesn't reject the graft. And the marrow that stem cells we infuse, it has to be in an environment where the stem cells can grow and repopulate, which is important when you're thinking about what you're going to do to prepare the patient. And this is also important too, is looking at the areas of distribution of adult marrow in the patient, because also this is the area where a radioisotope with an antibody will be targeting. So transplant, why do we do it? We do it because, as I mentioned, it's a curative option for patients with leukemias, myelodysplasia. But however, based on the status of transplant, the majority of patients that are transplanted are in a remission, because if we transplant patients with overtly relapsed disease, they will quickly relapse. Despite being in remission at the time of transplant, the most common reason of failure for a transplant is relapse. And this is just a pie graph looking at the reason why patients die beyond the first 100 days after transplant. And this is looking about 3 years out, and 59%. And this is national data with CIBMTR registry data. It's the 3-year mortality. 59% will die related to their primary disease. So disease control is a very important component for success of transplant. The use of radioimmunotherapy for transplant conditioning is to reduce late toxic effects that you get from external radiation, such as total body irradiation that we've been using in transplant since the last 40 years. And also, in theory, perhaps maybe a reduction in secondary cancers. And the theoretical effect of an alpha emitter is the cells that, the dose that the cells get, the cells are unable to repair, and it's a lethal effect on the cell, so the cell won't later on become develop a secondary cancer. And in theory, we'd like to be able to give higher doses of radiation without increasing toxicity for both disease control and also targeting the immune cells, so we get better engraftment of donor cells. And the antibody will target the radioactive isotope to specific cells, either malignant cells or T cells, in the case of immunosuppression. The target I'm going to talk about, give some results today, is CD45. It's a stable surface expressed antigen expressed on most leukocytes, which are white blood cells, and it has a high copy number per cell. And the majority of leukemias and myelodysplastic marrow-based diseases express this. And for the most part, it's absent from non-hematopoietic tissues. In the past, we've have a long history of using radioimmunotherapy. Initially, we worked with I-131 labeled on the same antibody with a little bit of chemo and a little bit of total body irradiation, showing that you can combine this and use it to condition. And actually, it's on the basis of the results of this study that the company Actinium has conducted a phase 3 randomized trial, and we're waiting results comparing this approach for transplant versus standard of care. And here I'll show that these are all patients that a majority of them had overt disease, and relapse still can be a problem, though you see long-term cures in a portion of patients that otherwise couldn't have come for transplant. Similarly, we did a small pilot study using Yttrium-90, which is another beta emitter with exactly the same antibody, showing very much similar results, though this was a small number, only 15 patients. Well, why do we want to do an alpha emitter? Part of the reason is the half-life of these other isotopes are longer. And if you think about it, if you want to use this in the transplant setting, you really want to target the marrow and the cells where the disease exists for a short period of time so that you can infuse the stem cells. Otherwise, you can't be irradiating the bone marrow. We also want to have something that has shorter path length, so you're limiting the radiation to the areas close to the targeted cell. So sometimes you can get some cross-target effect on other cells within the bone marrow or peripheral blood, but you're not targeting deep tissue into liver and kidneys or any other organs. And we like the fact that astatine has a high energy. Interestingly, people have concerns that this has such a short half-life. I did do studies initially with bismuth-213 off of actinium, and that has a much shorter half-life of 45 minutes. So this 7.2-hour half-life to me seems quite reasonable, if not ideal, for use in blood-based cancers. Initial studies using looking in mice, just looking at the effect of the radiation, you see very quickly, this is bone marrow. Looking at 24 hours, you can see that already, you can see that the marrow cells, it looks pretty lacy, and by 48 hours, most of the cellular component has disappeared or is diminished. So the effect is very rapid as compared to if you do total body irradiation controls. The decrease in cell number is much slower, though there is regeneration by 4 weeks. And this is the same as in spleen, other lymphoid follicles that you see here decreasing in size. And by 48 hours, the spleens themselves are about 25% of normal size and weight. Though again, the lymphoid nodules regenerate by 4 weeks. And this is the murine anti-CD45 antibody I should have mentioned. We did initial studies looking in transplant-treated normal dogs with just the isotope itself, did dose escalation, and determined that it was sufficient to get a graft in from the equivalent of an HLA-matched donor. It would be a donor leukocyte antigen identical littermate transplant. And the animals grafted quickly, didn't require a lot of transfusions. And if you see in the line with 200 centigrade total body irradiation, which is the bare minimum to get a graft in, you don't see as deep a decline in lymphocytes, or other counts, but they also regenerate quickly compared to when you give external beam irradiation. And there's also the bismuth superimposed with a dotted line. And we did some alpha camera imaging, looking at, and this is now in dogs, looking at marrow, where it goes. And this is panel C is bone marrow. And you see it lights up, it goes to the bone marrow. And also panel B is a lymph node at 2 hours. and panel A is a lymph node at 19 hours, and you can see it is very lacy. So it quickly gets rid of cells and lymphoid and marrow spaces. So to translate into the clinic for first in human studies, we need to be able to, and this is Yawen Li's and group with Scott Wilbur really did all of this part of the figuring out how to do the GMP production. Well, we basically how to scale up the radio labeling. We had to figure out also the GMP production of the CD45 antibody BC8, which is conjugated to B10. And then we really, due to our top-notch team and experience, when we submitted the IND for first in human studies, we're able to get this approved within the 30 days. So that's kudos to everybody that worked with us on this. And then similarly, we're able to get it through our IRB relatively promptly based on this, though we still had some logistics to work out. And basically, it's for anyone who works in clinical trials, there's a lot of stuff unrelated to the fact that this is first in human that gets a protocol online and ready to treat a patient. For specific to this trial, we had to work on the release criteria testing and the timing. And we also had to figure out the timing of administration to the product to the patient because of the fact that it has a shorter half-life. And the first patient was treated in November 2017. And this is just a cartoon of the production. You heard all about the alpha radiation of the bismuth target already. And just so that you understand, the other part that's happened is the B10, which needs to be put on the antibody, which is produced at the Fred Hutch Biologics facility. This product is made and vialed and ready to label. And it's on very early hours. The team comes in to label the target, and the radio labeling is done on the same day the patient is to be treated. So the objectives of the protocol talking about is to estimate the total dose of radiation to be delivered, the maximum tolerated dose delivered of the astatine BC8, B10 when given in combination with the other conditioning in high-risk patients with leukemias or MDS. We're always interested in this, the disease response. And also, we want to make sure that the patients would engraft, that we wouldn't cause some sort of marrow damage, that the patients wouldn't be able to engraft. And inclusion criteria, really, it's the most important part is that the patients were high risk for their disease, and they couldn't have circulating blood blasts higher than 10,000. This is a standard, all these criteria are pretty standard. They just had to have decent function and their kidneys and liver, and they had to have a suitable donor. Simplistically, this is the treatment schema. We give the radiation here, astatine-211 on day -7, and then we give our standard low-dose conditioning regimen of fludarabine and 2 to 3 gray total body irradiation, followed by infusion of the stem cell. And this conditioning regimen of fludarabine and total body irradiation would only be used in a patient that's in remission, not like the patients in florid relapse that we were planning to enter in our trial. So here's the first 20 patients. We've treated over 40 so far, but here's the first 20 patients, just so you get a flavor of what sort of patients they were. And probably I'd just like you to focus on the areas that are circled in red, where you see the percent bone marrow blasts. In general, we at the time of transplant would prefer if it was 0% marrow blasts at the time of transplant. And we even get concerned if it's 0.03 or in 3.5% blast, because these patients have a very high risk of relapsing with their disease, upwards of 40 to 50%, even when they have under the bar of 5%. So here you're seeing patients have refractory disease going into transplant in these areas where you see them circled in red. So there's very few patients that have 0% marrow blasts at the time of transplant. These are older patients, median age of 60, and 3 of them had failed a prior transplant. Here's the blood PK in the first group of patients. You see it's cleared very quickly by the first day after transplant. And this is percent injected dose per kilogram. And one measure is some of the patients had circulating peripheral blasts at the time of transplant. And here's a semi-log scale. This is the total number of blasts in the blood. And they got the astatine on day -7. So you can see the amount of blasts in the blood. And then with only one exception, they had it cleared even before they got their first dose of chemotherapy. So that's a measure seeing that we're able to clear the blasts out. And the one patient in the light blue line was our initial patient who got the lowest dose of 85 microcuries per kilogram. And what I didn't mention is these are single dose escalations where if there's no toxicity, the next patient could go up, assuming that it's been at least 30 days since the previous patient had been treated. And so a summary of survival in the first 20 of these high-risk patients is that the survival looks like it's about 35 to 40% long-term. And with additional patients, this looks similar. A certain proportion of patients did relapse, and some patients died of complications, which is not unexpected in this high-risk group of patients. Now, not everybody has a perfectly matched unrelated donor. As many of these patients are older, their siblings can't be used for donor, and so we have to look for other options, the unrelated donor registry. And for underrepresented minorities, because the unrelated donor registry tends to be Northern European, the likelihood of finding a matched donor declines. Whereas if you're white, you can find a fully matched donor in close to 80% of the time, whereas if you're an African American person or Pacific Islander, it's much less. So because of that, we've developed a trial where we're doing haploidentical transplants, like you can get a transplant from your child if you're older, or a parent if you're younger, or a brother or sister that's only partially matched. And this study is also concurrently being done, where the astatine now is given day -8. It's very similar, just a little extra dose of cyclophosphamide, which is another chemo that's immunosuppressive. And so that study is also currently ongoing. What I didn't put up is also we have another study for patients with sickle cell anemia, where we're doing a similar backbone. And again, these patients are hard to get grafts in because they're heavily transfused. But if you give them high doses of radiation and chemo, they do not tolerate this. And so we're exploring this in this patient population, and we've treated 2 patients there. So to summarize, the preclinical data and early clinical data show that the radioimmunotherapy is well tolerated. We can deliver supplemental doses of radiation to leukemic sites, and this anti-leukemic potential may improve post-transplant outcomes. And these encouraging results support more radioimmunotherapy-based transplant or other cellular therapy trials. Another targets will allow therapy in the non-transplant setting, which we're looking at in the preclinical animal models. And so this is just a subset of the different funding sources that we're using, primarily from NIH, looking at the clinical translation using Astatine-211, as mentioned, a lot of it in our malignant diseases, non-malignant diseases. We're also targeting CD4 and CD3 for models of simian HIV. And Damian Green, as Yawen mentioned, has 2 trials targeting CD38 for multiple myeloma, along with some other studies. And of course, when you sort of think about this, it really is a team effort that is patient-centric, that brings together such a large number of people in order to be able to do this. And that is it. And I look forward to questions during the questions and answers.
Thank you very much, Brenda. That is very impressive presentation. And I see there are questions for you in the Q&A right now. We will hold that question and because we have one more presentation. So let's move on to the final presentation of the day. Dave, please.
Well, great. Good afternoon, everyone. We appreciate the opportunity to share our work with Astatine-211 with the group. This has been a fantastic session today and a lot of great information. We're going to come at this from the commercial side, industry side of production and supply of Astatine-211. So we'll bring it back a little bit more technical for our presentation this afternoon. So let me start first with a quick introduction to Ionetix. For those of you who may not be familiar with us, we are an isotope producer and radiopharmaceutical manufacturer. So again, we're coming at this from the industry and commercial side. The company was founded in 2010 when we licensed accelerator technology from MIT Plasma Fusion Center. And that's really core and central to Ionetix. It's our in-house ability to design, engineer, and manufacture cyclotrons. But we're very use case driven. We leverage that cyclotron technology ability to look at where there's supply needs in the market. And then we deploy those cyclotrons. We own and operate themselves. So we're developing a network of operational sites to supply a specific isotope or a radiopharmaceutical. And so our accomplishments over the last 12 years, we've engineered, designed, and now we're manufacturing a 12 MeV cyclotron for PET isotope production. We're now working on a 30 MeV cyclotron for alpha isotope production. We have a network of operational sites in the US, and we're bringing online an alpha isotope facility. We have an ANDA for N13 ammonia that we use for our 12 MeV cyclotron. And we have an outstanding exceptional inspectional history with the FDA. So we've completed 8 successful FDA inspections. So that's a little bit of background to Ionetix, and we're certainly coming at this from a completely different perspective than much of the previous presentations here. But I think we can all agree that there's growing interest and demand in targeted alpha therapy. And I think Yawen really positioned nicely the advancements being made in our opening remarks with Astatine-211. And we see that, we see the demand by researchers and the growing need to increase the accessibility and availability of Astatine-211. I think all the alpha radionuclides have their own unique supply challenges to increase their accessibility and availability, and Astatine-211 is no different. I think we all know that in order to produce Astatine-211, you need to accelerate alpha particles up to 29 MeV and hit a bismuth target. And therein lies the challenge for widespread production and availability of Astatine-211. The commercial infrastructure of cyclotrons in the US and really globally are all predicated on proton machines in the 12 to 18 MeV range. The cyclotrons with the specifications to produce Astatine-211 are really relegated to our academic research institutions, and thus, a lot of the university work that's being shared with this group. And if you look at where these institutions are in the US, they really book in the US. They're very coastal oriented, right? And I think you can add NIH to this list. And so we're really stepping in as a commercial provider to develop a supply of Astatine-211 to help promote the clinical development of astatine and eventual clinical use of astatine labeled targeted alpha therapies. And so we're headquartered in Lansing, Michigan, and that's where we're going to start with our first facility. And that really fills a void in this university production network of within the US, we're going to really fill that central US void of access to Astatine-211 when we bring our facility online in Michigan. We're using an existing cyclotron technology that we're upgrading to produce Astatine-211, and I'll touch on that here in a minute. But our long-term plans are to develop our own 30 MeV cyclotron, put that into manufacturing, and just as I alluded to in our introduction, that's our mandate, is to look at ways we can increase the supply and availability of Astatine-211. So we plan to decentralize our production of Astatine-211 and creating regional production sites over time, again, to extend the reach and availability of Astatine-211 as these programs develop clinically and move into drug development and hopefully clinical access for routine treatments. So just to start with our facility in Lansing, Michigan, this is a freestanding commercial facility that's going to be dedicated to alpha isotope production. Not shown on this diagram here is we're already putting an addition to this facility. We're going to extend out the back and add a second vault. We're going to add another 5,000 to 7,000 square feet and add a second cyclotron next year to this location. And then forward of the cyclotron is our radiochemistry lab. And in the radiochemistry lab, we'll have full resources to process targets, to isolate and separate the Astatine-211. We can ship out Astatine-211. We can also, we'll have clean rooms where we can radio label the final drug product. And so at our facility in Michigan, we'll be able to do all the above in terms of producing astatine and supplying the isotope or the final drug product to interested researchers or pharma companies. This facility will be CGMP compliant and as well as Part 211 compliant for drug manufacturing. So the cyclotron is on site. It's been stacked and partially assembled. So we've been able to stack the magnets, and we've vacuum tested the cyclotron, and then we've mapped the magnetic field. So we brought this online, did a complete map of it, shimmed the magnets accordingly, and that wrapped up in May. And now we put the vault around the cyclotron, and that was completed about 2 weeks ago. So we have 6-foot concrete walls surrounding the cyclotron now. We need a few more weeks for the concrete to cure, and then we'll be able to seal it, and our engineering team can get back on site and start installing the subsystems. We'll eventually move to beam testing and full commissioning. So with respect to our facility in Michigan, the cyclotron will be fully commissioned by the end of December. The radiochemistry lab will follow that. So it'll be qualified and commissioned by March of next year. And that puts us into astatine production and distribution in April of 2023. We are planning to file a drug master file for Astatine-211, so that DMF will be referenceable no later than July and possibly as early as late April or early May. And if you follow Ionetix, you would have seen our press releases. We have signed contracts to support the clinical development of Astatine-211 targets. We signed an agreement with Precision Molecular and Atonco to support their development of their Astatine-211 targets, both of which should be IND phase 1 ready early next year. And so timing aligns nicely with bringing our facility online for production and supply and supporting these programs. Of course, we're in discussion with several others. There's good interest around Astatine-211, and we hope to obviously add to this list over the coming months. So I'd like to spend just a few minutes talking about the cyclotron technology, right? This roadmap to an Astatine-211 commercial production cyclotron that can support the long-term growth and availability and accessibility of Astatine-211. So for us, it all started in 2019 when we got a call from Duke, from Dr. Zalutsky, to have our engineering team look at their CS30 cyclotron that was being used to produce Astatine-211, or that is being used to produce Astatine-211. And so our engineering team was contracted by Duke, and we completed those upgrades. We learned a lot about the CS30 cyclotron. We learned a lot about Astatine-211, the value of it, the promise, and the need for additional production solutions. And so we made the commitment to start an alpha facility in Michigan following that work with Duke and for Dr. Zalutsky. And so for us, then it was a question of what cyclotron do we put into operation? And it was really a matter of how quickly we can bring on production and supply. And so we looked around, we actually found a CS30 cyclotron that had been decommissioned. This was a cyclotron that was bought and sold to the University of Michigan, just down the street from us. It was operated there until a number of years ago when it was decommissioned and put into storage. And so for us, upgrading that CS30 cyclotron, we know the CS30 cyclotron works well, has good performance for astatine production from our work with Duke. And so for us, upgrading that cyclotron would allow us to put it into production much sooner than if we engineered and designed and started manufacturing a 30 MeV cyclotron ourselves. So that is indeed what we did. We have completely rebuilt this cyclotron from the ground up, and we've upgraded the performance. So all the obsolete technology has been replaced with current generation technology. All the age-related components have been remanufactured and replaced. This is essentially a pretty much a brand new machine. And then we did upgrade several of the components, and I'll touch on that here in a minute. And so moving forward, our engineering team now is working on an Ionetix 30 MeV cyclotron that can accelerate alpha particles, and that will allow us to eventually put that into manufacturing and deploy those more regionally so we can decentralize production and increase access again for astatine over time. So we're going to have the Ion 30 machine that we'll put into manufacturing, then we'll have an upgraded version of that, which will be a high intensity machine. We'll do an external ion source. So just from a specification standpoint, the CS30 is the top row, and that's really baseline for us. It's a good machine. Again, it's what Duke is using. In Michigan, we're going to call it an upgraded CS30 cyclotron because we've reworked the central region, we've reworked replaced the sectors, the magnet sectors. And then from there, our Ion 30 machine, we're going to move to a 4-sector machine for better beam focusing. And then we'll have, again, a high intensity machine where we'll go with an external ion source and crank up the current that will allow us to do more from a targetry perspective and even extract the beam. The CS30 and our upgraded CS30 and even our Ion 30 machine will be an internal target for astatine production. So just to touch real quickly on the upgrades that we completed on the CS30 machine, our engineering team looked at the CS30 system and did a complete electrical and magnetic field analysis of the system and looked at ways we can improve the performance. And the first change we made was to rework the central region and the polar, and that was to improve the beam capture efficiency of alpha particles. And the more beam we can capture into acceleration, the more we can deliver on target, and again, improve the performance. And then the second was to look at the magnetic field map of the CS30 and look at ways we can improve the delivery of those alpha particles to the target. And the first thing that we found was that the CS30 had a very strong first harmonic. And when you look at the isochronous curve for accelerating alpha particles, there's a lot of errors in that beam transmission, which relates to a lot of beam loss. And so our engineers reworked the magnet sectors. We reduced that first harmonic, and then we were able to pull in those alpha particle accelerations into an ideal isochronous curve. And that's going to improve the performance of it. It's going to improve the beam transmission from the ion source all the way out to the target, where we can really control the beam spot and distribution onto the internal target for astatine production. And then the other upgrade on the CS30 was because it's an internal target system, we reworked the target control arm and put some more degrees of control and rotation on the beam head. So when we insert the target into the beam line, we can actually rotate the angle to maximize the beam distribution onto the target face. We're using a very similar target that Duke is using. We made some minor modifications to it. We eliminated a curvature in the channel for the bismuth, and we changed a little bit of cooling fins on the back, but otherwise it's similar target to what Duke is using. So just in summary, we've got our new facility coming online in Lansing, Michigan, where we have an upgraded CS30 cyclotron that will be starting astatine production and delivery in April of next year. We expect to produce on the order of half a curie of Astatine-211 per day. So we expect a strong performance out of this machine. We can produce and supply the isotope. We can radio label and produce supply the final drug product. And then again, we'll use our experience there to decentralize that process and put it into manufacturing our Ion 30 machine over time to increase the availability and accessibility of Astatine-211. And with that, I'd like to acknowledge our cyclotron team led by John Vincent. We have a world-class cyclotron team assembled, most of which who came from the Michigan State University superconducting cyclotron lab with 20, 30 years experience. Again, our cyclotron team is core to Ionetix. And then our operations team, we've got Alex Yordanov now heading up radiochemistry for us in our work with Astatine-211. And that concludes my talk. I thank you for, again, the opportunity to participate, and happy to answer any questions in the Q&A session.
Thank you, David, for the great talk. I'm very happy to learn about the status of Astatine production in your company and where your plans for moving forward. So let's all the presentations are completed so far. Let's move on to our Q&A session. I see several questions related with clinical studies. So first one, what is the age population for Brenda? What is the age population that can accept the target alpha therapy for AML? I think the current medicinal treatment for AML also has 30 to 35% survival. Have the patients recruited to these trials already taken other medications that did not work before beginning the radiotherapy?
Yeah, thank you for the question. Well, with AML, basically people can be put in 3 buckets. One is considered standard risk, and then intermediate risk and high risk. Standard risk would not go to transplant after standard chemotherapy. They would only go if they relapse with disease, which would happen 20–30% of the time. For intermediate and high risk patients, once they get into remission, we would take them to transplant with standard conditioning with either chemo only or chemo or low dose TBI. So the majority of patients on this trial are those who don't fit into getting a standard conditioning regimen because they come in with relapse disease. Because for leukemias, essentially with rare exception, they would have to be in a remission. And so these would be patients that failed other treatments in general, or we feel they just can't get into a remission otherwise. And the age would be the age of transplant. Most of the time we don't put someone into a transplant over age 75, though rarely if they're really good condition. So in general, patients can be up to age 75. Which is generally, a lot of places across the country wouldn't transplant patients over age 70. My other hat I wear is, develop what we call reduced intensity conditioning regimens, because when I first came in the field of transplantation, many years ago in the 80s, anyone over 40 was considered almost too old for transplant. And in the 40s, those were really old people, we thought. And as we get older, we're transplanting older patients. We figured out how to do it better.
Thank you, Brenda. And there are a couple more questions for you. Can you elaborate on your HIV treatment strategy? And the other one is, have you noticed a change in the cell population during regeneration?
Yeah, for the HIV, that's one's a lot more experimental. In the field of HIV, the ART therapy, the antibiotic therapy to keep the viruses in control is very good. And but one of the issues is the long-term effects of patients being on the treatment. So a lot of the approaches in the HIV world is trying to get rid of the reservoir of HIV positive cells. And the reservoir primarily is in CD4 cells. So a lot of people have done some studies using naked antibodies, and we have thought, well, if you use naked antibodies, they're probably not going to get rid of the cells. And so we thought, well, how about if we use And some people are doing antibody drug conjugates, which are another way you arm it with a chemotherapy. And so we're taking the approach of doing radioimmunotherapy, and it's still very much in a preclinical situation, and it's a work in progress, but really it's targeting the reservoir of HIV where the cells hide out. And we've done those studies, did initially some study in mice, and we're doing the work in non-human primates. And then the other question, sorry, Yawen, you're muted.
Oh, sorry. Have you noticed a change in the cell population during regeneration?
The difference that I notice, and we've done a lot of work in the preclinical dog model where they're perfectly normal. They haven't been treated with chemo. They've only received the radioimmunotherapy, is that you get very quick regeneration of when you put the donor cells in, very quick regeneration. And that's it's the homeostatic drive that happens in the milieu of ablating the previous marrow. But the hematopoiesis just follows the same general pattern, other than it seems to be a little bit quicker, but also because you have ablated also the cells a little bit quicker. And one other thing I noticed in the chat section, the comparison of, concerns about what about actinium-225 versus astatine-211. And it depends upon what your application is. I think actinium-225 is probably superior if you're targeting a solid tumor where you want to have continual radiation dose. But for some of these blood containing cancers, such as the leukemias, I think it's a little bit to the detriment, because then you end up being limited by the long half-life. It's a gift that keeps on giving longer than you may want it for blood cells like platelets and a lot of the blood cells, just from because you do get some generalized radiation, and you know that if you do an irrelevant antibody, as it circulates, it does give some radiation, and a lot of the blood cells can be sensitive to that. So if you have a really long half-life, then I would be a little bit concerned and when you're targeting bloodborne diseases. Not saying it can't be done, but it's just an area of caution. And in particular, a transplant, it makes the logistics too hard to want to put in the healthy marrow stem cells from the donor if you have to wait till all the radiation is gone. You push it back. That was the problem we had with I-131. We had to wait, and the patients were basically without counts waiting to put the stem cells in because the radiation had to be eliminated either by half-life or eliminate from the body. So.
Thank you, Brenda. Thank you for weighing in on that question, the comparison of actinium and astatine question.
Speaking as a clinician.
Yeah, we want opinions from all different perspectives. And I also wanted to hear, maybe have Bob comment on this question, because you said you were planning to do PSMA and PARP1 clinical trials with astatine-211. What is your reason for choosing astatine-211 over actinium-225?
Yeah, we're going to do MABG. That's a natural, because Dan Prima here at Penn was. Sorry,
my mistake. Yeah.
Yeah, he had a large role in getting MIBG approved. So the MABG is a natural progression for him to do clinical trials with that. With PARP1, again, we think astatine-211, the 7.2 hour half-life, is perfect with small molecules. I think if you have a good F-18 PET radio tracer that has a good tumor to background ratio in a solid tumor, then it's a great candidate for labeling with astatine-211. We like our PARP tracer because it's PARP1 is an enzyme that's right adjacent to DNA, so you're you're delivering the alpha particle exactly where it needs to be on the most radiosensitive organelle within the cell, DNA. So we think all the stars are aligned with our PARP radio tracer for doing therapy studies with Astatine-211. But yeah, I think for actinium, again, because of the long half-life and the comments that Brenda mentioned with respect to the, the types of, how it can be used with, it probably works well with the antibodies, but it doesn't work well with small molecules. And I'm a small molecule person. So I like Astatine-211.
Thank you, Bob, for your comments. There is another comment in the Q&A session that says, in general, tissue distribution of antibody is lower or slower than small molecule or peptide. Half-life of Astatine-211 is short. I'm concerned with that drug delivery to target site and the effect of alpha emission within short time. So yeah, I will turn this comment to Brenda again.
Yeah, that is correct, that there is a concern about that, which is why, well, for the small peptides or small molecules, that's not an issue. People are looking at that. And also you can do pre-targeting, where you first give the dose of the antibody part to the patient first, and then give the isotope later. As you know, Yawen, there's that's what Damien Green's group is looking at
Yes.
in this,
Pre-targeting,
is
yeah.
to do pre-targeting, which is why I guess generically in my mind I was thinking antibodies when I made the comment of for solid tumors that perhaps that's where you need the actinium. Though I must say, like into the lymph nodes, you do get percolation in by the 2 hours, and you do see the antibody in there. I didn't show the micrographs, but at least in the preclinical dog studies, antibodies were able to go in. You can use smaller molecules again to target if you're playing astatine. And I think you have to, it's not one is, you have to target, or target's a bad word. You look at the clinical scenario, what your goal is. If you want radiation to be delivered a little bit more long-term, actinium's probably better. And if you want to go in, do your damage and be done with the isotope, astatine would be better. And for a small molecule, I agree that you probably want something shorter acting than long acting. You sort of match the clinical scenario with what you use. I see room for both.
Yes, definitely for the blood-borne cancers that we are looking at right now. The targets, the antigens are so accessible. So the kinetics of the antibody is not an issue in this specific scenario. Okay, so I will. Let me see. There is a question for David. What is the mass of bismuth metal in your target?
Yeah, so again, we're using a similar targetry than that's used at Duke. So it's going to be 80 microns thick, and I believe it's somewhere on the order of 100, 200 milligrams of bismuth.
Thank you. So I'm gonna pick questions that are first that are directed to a specific panelist first, and then we will move on to those questions that are more like general for discussions. So Lauren, in your talk, slide number 15. Thanks for your presentation, Lauren. You have shown capability of different extractants on slide number 15. While they show good extraction capabilities for Astatine-211, I was wondering how well Astatine-211 is back extracted from these solvents. Also, how about FDA approvals for use of these solvents in medical applications?
Okay, so the 3-octadone, which is what we're currently using for extracting astatine, is an FDA-approved molecule, and that's why we've been using it. The back extraction is, so we're doing the back extraction is not an issue because we're eluting off of the column right now. And so we haven't looked into that. So.
And FDA approves, I'm sorry, you know.
Oh, on the other molecules, I don't know the details of all of that. And we're not thinking about using it. Normally, we're just looking at the fundamental chemistry right now. So I don't know the details. I'd have to look that up.
Thank you. I don't know either, but it's something, you know, good to think about. So again, this is for you, Lauren. Has any small molecule, a peptide or a protein been radiolabeled using Astatine-211 harvested by this method? What is the radiolabeling yield and what is the Astatine-211 species existing in the ethanol alloy from the column?
Okay, so there has been a small molecule has been labeled at MD Anderson using a copper mediated radiolabeling approach. And that is an ongoing thing. So that's what I'm going to say about that. I don't have the radio labeling yield right in front of me, but if whoever this is wants to tell us who they are, we might be able to tell them later. Since this is an anonymous question. And the species existing in the alluate from the column is astatine speciation is a complicated question. So I don't have a tidy, quick answer for that. That's something we're looking into all the time, trying to figure out exactly what's going on with speciation so that we know what's going on with our chemistry. So you want to reach out, we can try to talk through that more, but speciation is a very difficult question with astatine particularly.
$1 million question.
Everyone wants to know, so if you figure it out.
Everyone wants to know. All right. Thank you, Lauren, again for answering the questions from our audience. So there is another question asking what would be the desired specifications for Astatine-211 being sold? So I will try to answer this one first, if and then have other panelists to weigh in if they have other comments. So in my opinion, it's really important that the astatine has a very, very high radionuclidic or radioisotopic purity. It's really you can produce astatine-210, which decays to polonium-210 and is known to be highly toxic for humans. So although I should mention there is polonium-210 is exists in nature, in the environment. So there is an ongoing or active discussion, like what would be the acceptable level of polonium-210 or astatine-210, acceptable level for that radioisotopic impurity. And for now, we will just avoid it completely. And it's also that the product has a high radiochemical purity. So we know we have a single species of astatine, and it can be used for radiolabeling, whichever radiolabeling method you decide to use. So those would be the two most important things that, in my opinion. So I would see if any other panelists would like to weigh in. Okay. So
can you repeat the question? The most important things with respect to what? I missed that.
Yeah, the question is, what would be the desired specifications for Astatine-211 being sold? So I just assumed that's asking about like quality controls, things.
I think the desired specifications that there be a lot more of it. Yeah,
Yeah.
I think it's limiting the entire field. I mean, it's limiting every aspect of Astatine-211 research, including using it application-based, because it's a very easy thing to criticize is why are you developing radio labeled molecules with Astatine-211 when its availability is really so small, so limited. So I think, increasing the availability is priority one in my mind.
David's going to fix that problem.
Absolutely.
Well, it's Astatine-211 is probably the easiest alpha emitter to produce as long as we put in the cyclotrons, right? So, okay, so this brings us to the question that what needs to be accomplished in the next 5 years to make Astatine-211 a viable radionuclide for targeted alpha therapy? So, to me, a lot of this can be learned from the process of F18 and actinium-225 or iodine-131, those, radionuclides that have become more like standard radionuclides now. Maybe actinium-225 is not standard yet. So we can learn from the process that those radionuclides that have become, from research to like a commercial or more widely used radionuclides. And like Bob said, availability is definitely, a key factor. And automation, like you want to have automated equipment for isolation and for radio labeling, so it can be standardized and meet all the CGMP requirements. So okay, if no other comments from the other panelists, I will close this question and move on to the next one. Do you think short half-life of Astatine-211 is a serious shortcoming for its development and commercialization compared to other alpha radio isotopes with longer half-life, such as actinium-225? So I think we've already touched on this question, so we'll just move on.
Yeah, if I could throw something out there,
Yeah.
again, now the production of actinium-225 is not exactly a walk in the park either, right? I mean, this is very difficult to make, and it was made a national priority, and that's why some of the barriers were overcome. I think, again, going back to the prior question, Astatine-211 needs to be made a national priority, and if once it's made a high priority, then these barriers will be eliminated, much as the way it was with actinium-225.
Yeah, because I agree, because when I was working with the bismuth, which is another reason why I switched over to astatine, was because we needed to get the actinium-225 cow to milk it to get the bismuth. We couldn't get production, and we're paying $50,000 for one shipment to use for a few animals experiment. And they're talking about giving money to Russia to keep the Russian nuclear medicine people out of nasty hijinks, and focused on that. So yeah, I think a little bit of money and ingenuity, astatine would be available.
Those are great comments. Thank you, Bob, and thank you, Brenda. There is one last question I see. What are the panelists' thoughts on the Astatine-211 generator? So yes, Astatine can be made available through a Redown-211 Astatine-211 generator system. There are actually active research in that area. I know a group at the Argonne National Lab is looking at that production route. I will and I think Michigan State, the FRIB project also has radon-211 listed as one of the radioisotopes they can potentially produce. But I would also say that the production of radon-211 is not trivial. It's also a challenge, and you are a very special facility, a very highly specialized facility will be required for making radon-211, and how to make it without the radon-2, I think it's the radon-210, right, and the impurity that also leads to Astatine-210. So yeah, it definitely, the generator system has its advantages. So I look forward to see progress in that direction. Then going back to the cost comment, how do you think the cost of Astatine-211 will compare to Actinium-225? All right, so the cost, I'm not an expert in Actinium-225 production. I know some basics about it. It takes a national national lab team to make that happen right now. I know companies are wanting to step in to increase the availability of Actinium-225, but I haven't seen any Actinium-225 produced from a private company coming out yet. As far as I know, the cost of Astatine-211 is actually quite low. The target material business is monoisotopic. You just use the natural abundance target material, and the cyclotron is relatively easy to build. And I would I think I should not comment. I'm not the right person to comment on costs. So I would direct this question to the NIDC.
I don't think Roy's going to let me off the hook here. I was trying to avoid that. The production costs for actinium and Astatine-211 are both really high, right? And that's taking it outside of governmental production and through the DOE supply, right? So the infrastructure costs right now to support alpha isotope production, regardless of if you're making actinium, where the radium material, if you can get it, is really expensive, or Astatine-211, where the infrastructure of cyclotrons is you have a lot of expense in the infrastructure itself, right? So there's going to be a period of time in which all these programs are starting to advance in the clinic, where the costs are going to be high just because of the low volume and the moving these programs into phase one and increasing their volume. But over time, I think Yawen brings up a great point. The bismuth is really cheap, right? So the production cost will have some efficiencies over actinium production costs long term. But it's really critical that we that we can move these programs forward, right? Get more clinical programs started and into human use, and advancing these programs where volume helps to manage the infrastructure costs that's going to be required to build out supply. And I think the takeaway for Astatine-211 is you can really blanket and increase widespread access to Astatine-211 with the deployment of infrastructure and cyclotrons to provide that market coverage, right? And that's obviously an area that we're focused on.
Thank you so much, Dave. All right, so we're a little bit over 12 Pacific time, 12 now, and if there is no further questions for the panel, I will thank our panelists again for their great presentations and for their sharing their input and answering all the questions from our audience. And thank you all for participating.